next talk. We have a presentation by Kimberly Schell. She is from Newcastle University, and she will be talking about serum extracellular vesicle miRNA profiling to determine extracorporeal photopheresis response in graft versus host disease. Kimberly, are you online? You can share your presentation. Yeah.
All right. And it should work.
Yeah, we can see your screen.
Yeah. Can you still see it now?
Yep.
All right. Let's see, how do I get rid of this? Sorry, I just need to I don't know how I can pop out the cameras to remove it. Ah, okay, I figured it out. Hello everyone, I'm Kimberly Schell. I'm a PhD student at Newcastle University, and my project is about serum extracellular vesicle microRNA profiling to determine extracorporeal photopheresis response in graft versus host disease. So first off, in the current day and age, for hematological malignancies, diseases, or deficiencies, one of the last treatments, and also often curative treatments, is hematopoietic stem cell transplantation. But while it is effective, about 40 to 60% of these patients develop graft versus host disease, and also about the same percentage of patients will die due to this complication. Well, there are two main forms of graft versus host disease, which both lead to an aberrant immune response from the graft versus the host, as it recognizes the body as foreign. They have different etiologies that this affects different organs a little bit differently, but the main thing is that the immune system is now attacking something that it shouldn't. And when we want to suppress the immune system, the first line of treatment is steroids, which is usually effective, but it has its own long list of unpleasant side effects. But there are also patients who do not respond to steroids. So with that, these difficult to treat patients or refractory patients will go on to receive ECP therapy. And then, sorry. As you can see here at step 1, this comprises of a pheresis step where he separates the buffy coats. So all the immune cells are then treated in step 2 with a photosensitizing agent and UVA radiation. And all of this together is reinfused to the patient with plasma and their erythrocytes. This leads to apoptosis of about 10% of the immune cells. And then the current consensus is that this tolerizes the immune environments due to intravascular administration of apoptotic host cells. And some of you might know that MSCs are also often used for this process. But in this case, it's a treatment that you can easily feed back into the patient, and only takes about 2 hours if you have an inline treatment. However, there's currently no way to tell whether people are responding or will respond well. And it takes about 8 to 12 weeks before they come to the first checkpoint to see whether treatment is actually effective. So this is very cost effective, and it takes a very long time. So for that purpose, we are looking for biomarkers. And previously, Crosland et al., which is my supervisor, she discovered that in acute GVHD, there are serum and extracellular vesicle microRNAs that are differentially expressed in patients who develop GVHD versus those who do not post-transplant. And such, I'm interested in looking at the small EVs in serum, which you can see highlighted on the left. And then additionally in microRNAs, because EVs are known to have their effective cell-to-cell communication and actually influence the cell by the surface markers that they carry, and also by their cargo, which are, my purpose, mainly microRNAs. Therefore, I received samples from the Great North Children's Hospital in Newcastle and analyzed the serum at different time points. So before treatment, 4 weeks in, 8 weeks, and 12 weeks. And these will be referred to as time point 1, 2, 3, and 4. And we had 6 complete response patients and 6 partial response. which are referred to as CR and PR. So received serum precipitated the EVs out, isolated the RNA, and then also concentrated it. And with these samples, we did size and concentration determination with nanocytes, used transmission electron microscopy for the morphology, assessed surface markers through MaxPlex Milteni kits, which has 37 markers in it, and then also measured the RNA concentration, and finally used the NanoString Encounter 800 microRNA panel. And it's important to note that this is for direct detection, and it is curated to look for biomarkers and the biologically most relevant microRNAs. And there's also a paper brought up by my supervisor where she optimized NanoString for very low inputs and has got picogram concentrations to work on this assay. And also that we use the positive control normalization according to the geometric mean of the top 100 most stably expressed microRNAs, as there are no housekeepers in EVs, as we all know. And then lastly, we use the DE-seq package to analyze this in R. So my aims are to identify biomarkers for ECP response for the advancement of personalized medicine for patients with refractory GVHD, and to further investigate the role of EVs in the mechanism of action of ECP therapy. And I will be doing this by assessing protein surface markers on EVs isolated from the pediatric patient's serum, as well as exploring the microRNA cargo held in these EVs. So for the characterization, for the size and morphology, you can see on this negative stain that there are several smaller EVs, and then that the expected cup shape is observed in the large EV in the center where it has dipped in. And then looking for the absence of a cellular contaminant, you can see that at about 90 kilodaltons in the EV lane, there is no calnexin, and in my cellular control, which is PBMCs, there is calnexin present, so no cellular contaminants here. And then lastly, derived from the MaxPlex flow data, you can see that all individual samples have expression of CD9, 63, and 81. Next, for the size, we looked at this modal size of all samples, but there is no significant difference between CR and PR, which will throughout the presentation be referred to as green and red. But when you look at separate time points, you can see that for PR, there's a significant drop off from time point 1 to time point 2, which is actually this one and this one here. But we do not know why yet. And then it is also displayed as a time course, so you can see that PR has a significant drop off of modal size of 180 nanometers to about 120 nanometers from time point 1 to time point 2. Next, for the 37 markers assessed by MaxPlex, you can see indeed again that there's tetraspanin expression, but also that there are various immune compartments that have expression across NK cells, T cells, B cells, the innates, but also a lot of platelets and endothelial markers that are expressed for the EVs, as well as cell signaling molecules. And it's important to note that there is no significant difference here, but we can see the cell type origin of the EVs assessed. And this was at endpoints only, and further time points still need to be analyzed by MaxPlex. So next, displaying this in a heat map, you can see that the partial response patients seem to cluster together quite nicely, and that a lot of them have upregulated not just the tetraspanins, but also some platelet factors. And then to display this in a PCA, you can see here that the complete response seems to cluster in partial response, and then there is a mix of the both of them separately. And then next, we can see here a plot of the driving factors, so that certain groups of response seem to be driven by, for example, mostly T cell or HLA, as well as some endothelial markers that seem to separate out. And again, here you can see the clusters of complete response, which are distinct from partial response, and then this mixed cluster. Next, we use the RNA for a NanoString and to do some quality control on this, and also to see if there was any correlation between the amount of RNA and also NanoString results or viability. So the yield did not differ for any groups at any time point. And there is also no correlation between the count, the particle count, so the vesicles or the RNA. But what is important to note that the binding density, which is supposed to be a minimum of 0.1, was made here so that all samples actually went through the assay properly. And then next as well, we can see that for the known concentration spike in, there is a linearity of significant amount along with the measured positive controls. So this means that the binding in the assay was effective. And lastly, for the ligation controls, also another measure of assay effectivity, we set a limit of detection with an average of the negative controls plus 2 standard deviations, which is medium stringency. We can see that the negative controls mostly fall under this limit of detection, and that also all positive controls are above this limit of detection. Next, moving on to the analysis that we got through DE-seq2 of all the microRNAs that were assessed using NanoString. You can see that there are different time points denoted by circle, triangle, square, and the plus sign. So, and then also the complete and partial response again. You can see that there in the center is a distinct cluster of partial response in this PCA plot. Next, clustering this, we performed hierarchical clustering and displayed the Z-scores of differentially expressed microRNAs. And these are found to be the driving microRNAs between the clustering that you saw in the previous slide. And most importantly, I'd like to draw attention to microRNA 23A3P, which you can see is all very upregulated, and then also 378 E, which seem all to be downregulated in this particular pure cluster of partial response patients. Whereas you can see that the others are a little mixed, but we would need more samples to determine better clustering and see whether these microRNAs show up differently in different response, or perhaps patients who had a different indication to undergo transplant or ESP therapy. So next, you can see that the log fold change of microRNA 23A3P was the only one that was downregulated significantly, but it was downregulated consistently across the board, as you can see in the previous slide, and all the other ones were upregulated. So next, this was an initial analysis with the 8 microRNA signatures, not accounting for whether they were up or down regulated, where I input them into miRDip, miRWalk, and Tarbase. And from these 3 databases, I tried to find the overlap and found 8 genes that were targeted by these microRNAs, which you can see on the right-hand side. Next, I took these and put them into Reactome, where you get pathways like this that will tell you whether there's whether it affects, for example, the phosphorylation of CD3 and the TCR zeta chains. You can see that there is a clear immune involvement or targeting of these microRNAs. So I found mostly with this preliminary analysis that it involves the immune system, so T-cell activation, antigen processing, type 1 interferon upregulation, some signal transduction, insulin growth like factor 1 receptor activity, as well as apoptosis, cell adhesion, and nuclear transcription. And it's interesting to note that since this is a treatment that revolves around apoptosis and also immune cells, that this is again found in the microRNAs targets. So in conclusion, I've assessed EV surface markers, and they showed they originate mainly from immune endothelial and stromal cells and platelets, but there is no significant difference between the surface markers for complete and partial responders. But in fact, the microRNAs that are derived from these EVs do show a significant difference. So that will mean that the processes in these cell types are altered. And then lastly, also that the differential expression was observed between CR and PR across all time points, because the time points did not significantly cluster, but the response groups did, which means that there is a potential for predictive and prognostic biomarkers for treatment response. Lastly, I would like to thank especially my supervisor, Rachel, and as well as Aisling, Matthew, and Andrew, and the patients and their families, the ECP team, and some colleagues for their bioinformatics support, and also my funders. Yeah, I'll now take questions. Thank you.
Thank you, Kimberly. Are there any questions from the audience? Okay, so Denise, would you like me to read the question?
Yes,
Or do you want to?
First, thanks for your talk. It was very interesting. I have some questions to the bioanalyzer that you did with your RNA.
Yep.
So basically, you used the Pico chip, which takes, which gives out the RIN values, right? Did you observe any, strange things with the RIN values, or were they normal as if it would be cells, cell samples, cellular samples?
No, we did not look at the RIN values because for sequences this small, you usually won't observe a RIN value that you can work with. They won't be consistent. So we just looked at the concentration because we knew that with so little RNA, this is mainly what we would focus on, and with such a highly sensitive chip as well, it was basically the best option that we had to determine the RNA concentration.
Yeah, okay. Yeah, because so we were using this also, and we found very low RIN values, which of course you explained why they are low, which is why we also decided to exclude them. But also when we worked with the chip, I found that the RNA concentrations which are measured can quite vary. Did you have like stable concentrations also if you measured replicates or something?
They were quite variable, and for some values that seemed extremely high, I actually ran them again and they were still consistent. So in my experience, and as also for my purposes, I trusted the concentrations that came out of this analysis.
And have you tried any other chip, like the small RNA chip? It's also for the Agilent bioanalyzer?
I know of it, but I haven't tried it yet, specifically also because from our NanoString outputs, it seemed to work regardless of the input level, and the binding density, as I mentioned, was above 0.1 for all of them, so the assay worked for all individual samples.
Okay, cool, thanks.
So Barbara, I can see your hand is up. So do you have a question? That will be the last question for the session. So yeah.
Yes. Hi, thank you so much for the amazing talk. I saw that you show that you have difference along the time. So have you checked some circadian rhythm markers in these patients?
Do you mean along this time point?
Yeah, because you show differences. So if you check some circadian rhythm genes, or for example, melatonin or cortisol for these patients.
We do have serum left, so we could check that, but I might not have been clear about this before, but the sequential time points are weeks apart, and treatment cycles are always started in the morning. So it should be around the same time of day that they receive this treatment or that their samples are taken.
Okay, thank you. And I don't know if you already mentioned, but how much volume do you have from serum? So the initial volume that you use to isolate the RNA?
Ideally, we use between 500 to 1,000 microliter, but there have been samples where we have more or less of it. And I've corrected, for example, the RNA concentrations, which I showed in these plots, to calculate it back to as being 1 milliliter. So in this one and this plot.
All right, so thank you, Kimberly. So we're going to move on to the next presenter, and I request Amir to